Thank you all for joining. We're just thrilled to have such a great program and really excited for 2 days, 2 half days of interactions together. On behalf of the Novo Nordisk Foundation Center at the Broad and with my co-organizer Simon Rasmussen, one more time, just in case anyone didn't see the email or the announcement, we had a last-minute cancellation for our first talk this morning, which would have been by Deborah Marks, but we're thrilled to begin the workshop now. This is now the beginning of the workshop with Anshul Kundaje. Anshul is a professor of genetics and computer science at Stanford University and has done path-breaking work, the interface of machine learning and biology. And it's such a pleasure, Anshul, to get to introduce you again, this time as the opening speaker of our workshop. Thank you so much for joining us, and I'm looking forward to your talk.
Awesome, thanks so much. I'm actually going to present almost completely unpublished work, which is totally fine. You can still record the call and put it out there. But I thought it'd be fun to discuss new stuff coming out of the lab. And so today I'm going to focus on sort of an interesting deep learning model we now have. The code is available. You'll see the link at the end. The paper is not yet out, but we'll be releasing it soon. But these are base resolution models of chromatin accessibility, deep learning models that map sequence to bulk and single cell measurements of chromatin accessibility. And you can use them for many interesting discovery tasks, including understanding of cis-regulatory syntax, which I'll describe in a few slides, and then also regulatory variant effect prediction. And this is work led by Anushri and Anna. Anushri is currently a graduate student in the lab. Anna was a grad student. She's now at In Vitro. All right. We're going to discuss regulatory DNA today. And as you know, these are pieces of the genome, sequences in the genome that act as binding sites for collections of transcription factors. Transcription factors are regulatory proteins that bind DNA in a sequence-specific manner. Transcription factors rarely act individually. They act typically in concert with each other. There's a complex combinatorial code that's encoded in the DNA. Transcription factors cooperate and compete also with nucleosomes, which provide structural stability to the DNA, but also have regulatory function. And then, of course, these nucleosomes are also modified in interesting ways. And collectively, this is sort of the architecture of the regulatory DNA. And we're going to focus on the sequence code, trying to understand how evolution has encoded regulatory logic in these elements, enable transcription factors to perform exquisite regulatory functions in specific cell contexts. And we're going to focus on mainly today chromatin accessibility assays. There are a number of different experiments you can perform to profile regulatory DNA. For example, ChIP-seq will allow you to profile a transcription factor binding genome-wide in a cell type of interest for a specific protein. So here's some ENCODE data for a variety of different transcription factors. But these are pretty laborious experiments, and it's pretty hard to do hundreds of transcription factor ChIP-seqs and thousands of cell types. So what you can instead do is you can potentially use other kinds of experimental tricks. You can use the TN5 transposase or the DNase 1 enzyme, which preferentially either nick open chromatin. These are pieces of DNA bound by proteins, or they can, like TN5 will insert sequencing adapters into open chromatin. And so when you sequence the library generated by this experiment, you get a track across the genome, and the coverage at each position gives you some sort of probability of any protein binding in the genome. So it's kind of a universal assay for protein DNA binding. That's not actually entirely true, but on a first approximation, that's a good way to think about it. There are many proteins that don't show up in ATAC-seq and DNA-seq. However, given that it's a universal assay and pretty fast and relatively cheap to do, it is a very popular approach for profiling regulatory DNA. But everything I'm describing today can be applied to other kinds of experiments too. But we're going to focus on ATAC-seq and DNA-seq. assays for chromatin accessibility. And we're going to use these experiments. We'll perform, by we, I mean not me, but we are a computational lab, but we will take data generated by fantastic collaborators, and we will try to use machine learning models to understand how DNA sequence is producing or driving this sort of beautiful regulatory profile across the genome in any specific cell context. So how do you translate this to a machine learning task? Well, one way to think about it is, given a chromatin accessibility experiment, you can think of it as labeling the genome, right? Every position in the genome gets some coverage from the sequencing experiment, and you can treat this coverage in various ways. You can binarize it. You can use some kind of thresholding, peak calling, and say, oh, here's a peak, here's a peak, here's a peak, and the rest of the genome has no peaks. Or you can represent it as continuous entities of enrichment. These continuous measures of enrichment are typically summarized at 100 base pair, 200 base pair resolution, peak resolution. And then what you end up getting is, you can think of like binning the genome into many little chunks, and you have millions of these chunks across the genome. Each chunk is associated with some sequence, and the chunk is also associated with some label coming from the experiment. That could be a binary label or a continuous label. And so that gives you millions of chunks with sequences and labels, and you can apply your favorite machine learning model, some classification or regression model, to map sequences to labels. People have done this for a very long time, and it works very nicely, and you can do lots of exciting stuff with it. But as assays have improved, they've gotten more and more higher resolution. So what I mean by that is, for example, if you take an ATAC-seq or DNase experiment and you look at some peak, 100 base pair resolution, and now you actually zoom in, look at what's happening at single base pair resolution, like the actual coverage of read starts at one single base pair resolution, you will often see really interesting morphology of the read coverage profiles. So, for example, some proteins will protect the DNA from enzymatic cleavage, and so they will create what are known as footprints, valleys and coverage profiles. And the precise, shape of these footprints and the coverage profiles, the morphology, have really exquisite information about the protein DNA binding interface. And if you summarize this into 100 base pairs into a single scalar, you lose all this information, right? So deep learning models so far have not taken advantage of this sort of structure. There are other approaches, statistical approaches, that have used these, sort of try to leverage this footprinting methods, that try to leverage this high resolution architecture, but neural network models so far have not. So what I'm going to present today is sort of a hybrid of the two, and I'll show you why it has an advantage over both approaches. Okay. So what determines footprint shapes? Let's just discuss that for a second. This transcription factor residence time, that is how long the protein sits on the DNA. If you sit for a really long time, you protect the DNA much longer, and you get deeper footprints, right? Like CDCF, AP1. GR, for example, has a very shallow footprint. Right? But there are many other factors. The affinity of the sequence motif that the transcription factor is binding to has an effect. Surrounding sequence syntax and cooperative binding has an effect. So it's not just the motif, but what's happening around. Chromatin state obviously has an effect, but also transcription factor concentration, and the elephant in the room, enzymatic preference. So often we think that the experiment is the ground truth, but the experiment is just an experiment. And it is a convolution of it's a confounding of two effects. There is the underlying biology that's driving the experiment, and then there is the experimental protocol that's sort of adding stuff onto it. And if you're not careful, you will not only learn the biology, but you'll learn all the other garbage that the experiment is throwing into the readout, right? The experiment is not necessarily the ground truth, and particularly for very powerful models, you have to be very careful with how you model the data, because otherwise you can quickly go down the wrong rabbit hole. And we'll talk about enzyme bias a lot today. Okay?
Anshul, a quick question. I'm going to paraphrase from the chat. How do you think about the overlapping nature of the windows when you talk about your base pair resolution in the 100 base pairs?
Yeah, so at base pair resolution, you can use whatever strategy you like. I'll show you in a few slides. We don't care about peak calling or binning or anything of that sort. You can just walk across a genome one base pair at a time. So you can have overlapping bins if you like, and everything's happening in a continuous space at single base pair resolution. So this whole exercise of peak calling and stuff literally doesn't matter anymore. Okay, so we'll talk about that. But
Right.
in the previous scenario that I showed, people are usually using partially overlapping bins, and then they're augmenting the bins. They are like shifting them randomly. They are reverse complementing them, but they are typically 100 to 200 base pair label bins, and then 1,000 to 2,000 base pair sequences trying to summarize information to 100 base pair resolution, peak resolution. Okay. All right. So we decided to change this a little bit. We had a paper in 2021 called BPNet, which was used to model chip exo and chip seq data at single base pair resolution. We made minor tweaks to it, but basically for chromatin accessibility. The way it basically works is you're again walking across a genome. You're taking tiles of sequence, but this time we're not going to just only summarize the coverage over that sequence. So imagine you have a 2000 base pair window, 2114 base pair window, and you one-hot encode it into one-hot encoded sequence. You run it through a convolutional layer. Convolutional layers are basically pattern detectors that scan the sequence. Think of them like little motif detectors. And then you pass the output of these convolution layers to additional convolution layers. And these convolution layers now combine the patterns from previous layers to more complex patterns. They keep going, right? So you can learn arbitrarily complex patterns. But the key trick is we tag on convolutional layers and dilated convolution layers until the model in the final output layer gets to see any output position that the model predicts at. Gets to see 2000 base pairs. So it can integrate information over the entire 2000 base pairs or any base pair in the genome. It's called the receptive field of the model. It gets to see 2000 base pairs, integrate information in different ways through the convolutional layers, and then transform the sequence through the convolutional layers into two outputs. The first output is very similar to what people do before, which is the log of the total coverage within the 1000 base pair sequence centered at the 2000 base pair window. Okay, so this is like a summarized count of the total coverage. It's just a raw count. There's no normalization of any kind. You'll see why. And then alongside the total counts that we try to predict, we also try to predict the distribution of those counts over the 1000 base pairs. So we have like a probability distribution over 1000 base pairs, and we are predicting n number of reads. We're going to take those n number of reads and try to predict using the probability how we distribute the reads over every position in that sequence. Okay, so two distinct entities. And the idea is that one entity models the magnitude of the signal over the entire 1000 base pairs, and the other entity models the shape of the signal. We kind of decouple these two. You'll see why in a few slides. Okay, so that's Chrome BPNet. Very similar to BPNet, it just has many more convolutional filters. Instead of BPNet used to have 64 convolutional filters, Chrome BPNet has 512 because you are trying to capture a more complex syntax than that for a single transcription factor. Okay, that's it. So we train this model on a subset of chromosomes, and we try to see how the model predicts on held out chromosomes, right, in the genome. So we are not predicting across cell types or anything of that sort. We are predicting within a cell type. We're just training on some chromosomes, predicting on other chromosomes. I'll explain to you why this is still a useful strategy, even though you have data on all chromosomes. Okay. So this is to show you what this looks like at single base pair resolution. This is a tag-seq and DNA-seq in the GM12878 cell line. It's bulk data. And you can see that once you train and you predict this on held-out chromosomes, the observed data and the predicted data look remarkably similar. This is a tag-seq, this is DNA-seq for the same sequence and the same cell type. And you already start seeing some interesting differences, right? The model exactly mimics what it should predict. It's going from sequence, it's predicting, and it looks exactly like what the data is supposed to look like. But the DNAs in the attack look very different at single base pair resolution. So that's confusing because they're supposedly capturing chromatin accessibility. At the lower resolution, they look very similar. Like you smooth the data and you look at peaks, attack and DNAs look very similar. Single base pair resolution, they look very different. So we can evaluate this, chromosome wide, so that I'm not cherry picking some example. And overall, on total counts, remember we have two outputs from the model: the total counts in 1,000 base pair regions across the chromosome, and then we have the shape of the profile. So if you focus on the total counts, the observed and predicted have pretty high correlation, 0.7. This is only in peak regions. We are not computing this chromosome wide. If we did compute it chromosome wide, you'd have a really strong spot at 0, and the correlation would look something like 0.9. That looks great, but we really care about the signal inside peak regions. There's classification of peaks against background, and then there is prediction of actual quantitative signal inside peaks. So inside peak regions, the correlation is pretty nice. It's decent, 0.7, pretty state of the art. And genome-wide, it's higher. It's about 0.9. Okay, so that we are good at total counts. But we also try to look at prediction performance on profiles. And we use the Jensen-Shannon distance as a measure of similarity between observed and predicted profiles. Remember, our profile predictions are probability distributions over the sequence. The probability at every position in the sequence. So you can match them up. There's an observed probability distribution, a predicted probability distribution, and we compute the JSD as a metric, as a distance between the observed and predicted. So the lower the better, right? So the blue distribution that you see here is the distribution of JSDs over all peak regions, okay, between observed and predicted profiles. So it kind of looks okay, but we need an upper and lower bound. So to try to get a decent upper bound, we take pseudo replicates. We just subsample reads from the same, mutually exclusive sets of reads. We create like replicates from the same sequencing experiment, and we look at the similarity between pseudo replicate profiles. And that's this upper bound right here. You can use real replicates too, but they are worse than pseudo replicates, and they will often look something like this. Okay, so we are actually pretty close to replicate concordance and even overlapping pseudo replicate concordance, just sampling reproducibility. That's the upper bound. And then the lower bound is, you can have two lower bounds. One is you can just shuffle the profiles and compare observed to shuffled. That's the lower bound, or predicted to shuffled. And you can also compute the average profile for all the peaks as a prediction and use that exact same average profile across the genome for all the peaks, and then see what that looks like, centering on this peak summit as a prediction. And that's a more sort of conservative lower bound, right? So you can see that we are very far away from either of the two lower bounds, and we are partially overlapping the upper bound. That's replicate concordance. So we're pretty happy. This is, it resembles what you see here. It looks very similar. This is saying that, yes, at least in shape, it looks very similar. So what can we do with this model? We've trained it. Well, the whole point is interpretation. The model, we now hope, is an abstraction of the experiment. It can take any sequence and map it to accessibility, whatever accessibility you gave it in that cellular context. And so now we can use it like an oracle. We can ask it all kinds of questions, and hopefully it'll tell us about the cis-regulatory code. That's the hope. We'll see how that works. So if you want to interpret the model, you can do several different tricks. The first trick is to take any specific sequence in the genome, and you want to understand the contribution of every base in that sequence to the prediction, right? So we use this deep lift method, which takes a trained neural network. It takes the sequence, it predicts the profile, and then through a backpropagation algorithm, it takes the prediction of the model and recursively decomposes the prediction into contributions of the neurons in the penultimate layer. And then through a dynamic programming approach, it sort of recursively backpropagates information. So it decomposes this layer's contributions into this layer's contributions and keeps going all the way down to the individual bases. And then finally, it provides an additive decomposition of the predicted signal over each position in the genome, each position in each nucleotide. Okay, so every position and every base in the sequence, even the unobserved bases, gets a contribution score. We're going to focus on the contributions of observed basis, but there's interesting stuff you can do on these contribution scores of hypothesized contribution scores of alternate basis at every position. But deep lift gives you effectively an additive decomposition at every position. So you think of it like a linear approximation of the prediction for every sequence. It's not like one linear model for the whole genome, but it's sort of like a linear model for every sequence. So that's kind of nice interpretation. Okay, so what does that look like? Here's again.
Could you say what you mean by a linear model for every sequence? What's the linear mapping that you have in mind?
So the idea really is that if you sum up the contribution scores,
yeah,
right, you actually get the prediction from the model minus some reference prediction. And the reference prediction is typically coming from something like, we take dinucleotide shuffle sequences. If you shove them through the model, that's sort of like a reference baseline prediction, right? And you're trying to predict the difference of the actual prediction versus that reference. So if you take that difference, right, and you just you sum up the scores, you literally add the values over all the bases, that addition is literally exactly equal to the difference of the prediction from the reference prediction.
Okay, great.
Right? So it is truly an additive decomposition, right? It's a nice additive decomposition, but it's every sequence gets a different additive decomposition.
Right.
Okay, so let's say I take, here's some observed data, here's some predicted data. These are the deep lift scores for the count head and the profile head. Now remember, we have two heads, right? In the output of the model, there's a count prediction and there's a profile prediction. And we can decompose the count prediction and we can decompose the profile prediction in terms of contribution scores over the sequence. So when we do that, yours sort of at low resolution looks good. This is at high resolution, this is at base pair resolution, zooming into that peak. You can see the observed and predicted profiles are again very, very similar, right? Really nice. They look like replicas of each other almost. So we can take this predicted data and then we can look at the count contribution scores and the profile contribution scores. And you see something very interesting. There's a beautiful CDCF motif right here. The rest of the bases are all suppressed, nothing much happening. And that looks great. So CDCF is driving this really nice profile. But if you look at the profile predictions, contribution scores, you see CDCF, but you also see a whole bunch of other random stuff. And this, this doesn't look like any motifs we've known before. So somehow the two heads are not agreeing. Something is predicting the profile counts, and something is predicting the profile shape. And for whatever reason, the model says that they're not the same. So you want to investigate this a little bit more. And we saw this across the genome. Okay, they didn't agree with each other. So what we did is we use another method called TF modisco. What TF modisco can do is compute deep sharp scores. We compute these deep lift scores across all the peaks in the genome. So we get these very nice contribution scores. And you might want to summarize all the patterns you see across all the sequences in the genome, right? So that's what modisco is. It's a motif discovery method that takes contribution score profiles from a whole collection of sequences and learns consolidated patterns that it repeatedly sees in all the sequences. So the way it works is, again, not that complicated. The exact algorithm is, in fact, quite complicated, but the details matter a lot. But conceptually, it's very simple. You compute these deep lift scores across all the sequences. You try to throw away unimportant nucleotides through the lens of the model, and then you end up with these little seclets. Seclets are contiguous subsequences with high contribution scores. And then you use a clustering algorithm. We use like community detection, Lowain or Leiden, which is also used for single cell clustering with a custom distance function designed for sequences with continuous scores on them. We call it the continuous Jacquard's distance. And it's a very convenient distance for this kind of these kinds of continuous sequences. And then we cluster them, we align them, and we collapse them into motifs. Okay. This looks pretty. So now we're going to use bodisco on the count head, on deep lift scores from the count head and the profile head separately. Okay. just to summarize motifs from both heads for that same model that we just saw. So we run this for the count head in GM12878. We get several 60, close to something like 60 motifs. Okay, and they beautifully summarize the biology of the regulatory lexicon of this cell line. This is a lymphoblastoid cell line, and you see all the classic players. You see RUNX, IRF, NF kappa B, everything AP1, everything makes sense. Looks great. You do the same thing on the profile head, and things look super wonky. Okay, you pick up motifs, but they are kind of mangled. You see, like this RUNX motif looks so beautiful right here. It has a much sort of richer GC content on the flanks. You see like strange sequence patterns, and then a bunch of patterns just dominate the entire motif landscape in terms of frequency of motifs. They all have this CAG, 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 CAG. How's going on? Or different kinds of motifs. And thankfully, if you stare at motifs long enough, you realize, wait, you know what? This looks like the sequence preference of the TN5 enzyme. I've seen this before. So you're like, okay, something weird's going on. The count head is learning motifs beautifully. The profile head is learning motifs mangled with all kinds of GC bias. And there's also this really powerful prevalent motif, the TN5 bias motif, that keeps showing up. It's orders of magnitude more frequent than any TF motif. So something's off, right? The model's sort of learning different things, and the profile and count head are not agreeing. So let's go to talk about TN5 bias. The elephant has shown its face, so we need to kind of do something about it, right? So different ways in which TN5 bias has been estimated. One is to take naked DNA and transpose it with TN5. And that gives you, some kind of random cutting frequency across the genome. If there were no bias, that would look like a uniform distribution. It definitely is not a uniform distribution. There is very strong bias. The other approach, which is also interesting, is this is naked DNA, so it's not in chromatin. If you take ataxic data in any single experiment, and you take peak regions and you throw them away, you take the rest of the genome, we'll call that background. The background is not driven by transcription factors, right? So you expect the background to be driven largely by TN5 bias, but the TN5 sequence preference in chromatin, right? So one is in naked DNA, one is in chromatin. Suppose they should hopefully do the same thing, but long story short, they actually don't. The naked DNA transposition learns a slightly different kind of sequence bias than in chromatin, and so if you try to use it to correct biases later, it doesn't completely correct biases. It corrects partial amounts. Okay, so people have used this before, and what you can do is, for example, you can just center a window at every cut site in the genome in background chromatin or in naked DNA transposition, and you can learn KEMA models, or you can learn position weight matrices, or you can learn dimer models, dinucleotide models. People have trained a variety of different bias models. They're mostly simple models, usually KEMA models, or they are position weight matrices or some version of that. And they look kind of like this, right? You see the CAG, exactly what we saw right here, the same pattern, right? So yeah, the profile head is learning bias like crazy. So how do you correct for this bias? Previous approaches take a statistical approach. They estimate these bias motifs. And with these bias motifs, you can now scan the entire genome, and you can get like a bias score, like the likelihood of bias at every position. And then you can use some kind of statistical model to adjust these bias tracks, these predicted bias tracks from the sequence model, the position weight matrix model, or the KEMA model, and you can do some kind of fold enrichment or some kind of statistical correction, and you get some corrected profiles, right, from the data. So we were like, yeah, let's just use that. People have done this before. Let's just take the predicted profiles and make PromBPNet predict on the corrected profiles, bias corrected profiles, instead of the raw profiles, because that should correct the bias, right? So we do that. We actually just substitute the raw profiles with the bias corrected profiles from two methods, state-of-the-art methods, hint attack and Tobias, which are these statistical correction methods. And we again train the model, we run deep lift and modisco, and we do a test. We again test what the motifs look like from the count head and the profile head. The count head looks fine, again, as before. The profile head unfortunately gets, I mean, it does get much better. You can see, oh, it's cool, it's starting to pick up stuff. But there are still TN5, partial TN5 motifs. You can see it's not a really long 20 base pair motif. It's a much shorter CA-CAG, CA-CAG, CAG, but they're still dominating. They're still kind of all over the place. So it's not fully correcting the bias. So what we do is we are like, you know what, let's just, the bias model is a position weight matrix, or it's a KEMA model. We can just fit a potentially better version, just fit a neural network, right? Why simplify it? If the simple version doesn't work, so let's use a more complicated version. Always start with the simple version. If it doesn't work, use a more complicated version. So we basically just do that. We fit a neural network to the chromatin background, right? So we take peak regions, we eliminate them, and we take the rest of the genome, and we fit a Chrome BPNet-like model, a very simple model, not a really big model, because the bias, we don't expect it to learn thousands of motifs, maybe collection of few motifs. So it's a small version of the Chrome BPNet model. We train it on background regions, not on peaks, okay? And then we run deep lift and modisco on those background regions. And if there was no bias, we would not be able to predict the background from sequence at all. But in fact, this background model has extremely high performance. You'll see in a few seconds. It can predict the background incredibly accurately, at least the profile shapes, not really the counts. And when you run modisco on the background deep lift scores, you end up with 7 or 8 motifs, okay? And one of them is the canonical TN5 motif, the CAG, CTG thing. But if you just align these 7 or 8 motifs, you actually see they're slightly different. They're slightly different versions of each other. And so what the neural network is saying is that the TN5 preference is not a motif. It's not a position weight matrix. It's a slightly more complicated model. It's kind of a mixture of position weight matrices, and neural network does a good job approximating that mixture and using it appropriately. So this is nice. We now have an improved bias correction, improved TN5 bias model in chromatin. And now we can use it somehow to correct the data. So how do we do that? Well, we do it in 2 ways. We have sort of a bias factorized model now. Sorry, not in 2 ways. We kind of have a 2-phase process of trying to correct bias. So we call this the bias factorized version of Chrome BPNet. It again starts with the sequence, but there are now 2 neural networks operating in tandem, okay? One model is the bias model that we've pre-trained. Remember, we pre-trained it on the background. So we've pre-trained it and we've frozen all the parameters of this bias model. This bias model now takes the sequence, pops it through the bias model, and predicts the profile, right, and the counts, okay? So it takes a sequence, and just from the perspective of bias, it predicts coverage and shape. And now we take we take a second model, which is also like a replica, which is the Chrome BPNet model I showed you before. But now what this model does is it tries to fit the sequence not to the observed data. It tries to fit it to the residual of the observed data minus the bias, okay? So the predicted output of the model is a sum of the bias and the residual, okay? So the idea is the observed data is a combination of 2 signals. There's the bias and there's the biology, right? The TF biology. We're going to decompose the bias from the TF biology such that when we fit the model, it's fitting it in these 2 sort of parallel paths. And once we fit the model, we can just disconnect this portion, and whatever this portion predicts is the true underlying latent signal minus the bias, right? So we just fit it in 2 phases, okay? Very simple, not too complicated. Just an additive model at the end of the last layer, but the additive model takes in the bias and predicts the residual with the sequence model, okay? When we do that, it works really nicely. This is the profile head. You do not see any TN5 bias, all gone, right? It looks great. And all classic motifs, and then now the count head and the profile head exactly agree. You see the exact same motifs from the 2 heads, which is nice. You can test for this bias. You can use a model as an oracle. So these are the TN5 bias motifs. You can take them and just shove them into random sequences. Just take a synthesizer sequence, shove the motif into it, the TN5 bias motif, and now push it through the model. We're going to push it through 3 models. The first is the Chrome PP9 model without bias correction. The second is the TN5 Bias model that we had trained. And the third is the bias factorized Chrome PP9 model, just the TF head, right? The deconvolved TF head, the one that's supposed to only learn biology with no bias. Now, if you take these motifs and you insert them into random sequences and you look at the predictions of the models, 3 different models, you see that Chrome PP9 without bias predicts a big spike. The TN5 bias model predicts a big spike, which is exactly the TN5 bias, which is exactly like the Chrome PP9 model without bias correction. The difference between these 2 models is this one is predicted only on background regions. Sorry, trained only on background regions. This one is trained on peaks and background regions. You can see that they're both learning exactly the same thing. All the TN5 bias motifs fire spikes. But once you do the bias factorized correction, once you deconvolve the bias from the TF model, you can see they predict flat lines. This tells you that the TF head, right? Sorry, the TF module, this module, is truly insensitive to these motifs, as you might expect. It's obvious, but it's a good sanity check, right? That's how we set up the model. It does what it's supposed to do. It learns motifs, doesn't learn the TN5 bias, and in terms of sensitivity, it is clearly insensitive to the TN5 motifs. So we can run this on many different cell types and we pick up all kinds of nice motifs. Everything's looking good, all right? So let me show you some, see how much time I have, okay. So let's look at some marginal footprints, okay? So what I mean by this is now we can use a model to study footprints. So we can again synthesize arbitrary sequences, random sequences, and we can insert this time motifs of transcription factors we care about, not TN5 motifs, right? So we're going to take NF kappa B, GATA, HNF4A, SP1, and we're going to insert them inside random sequences. We're going to take 3 different models from 3 different cell types. GM1287H should be sensitive to NF kappa B only, whereas K562 and HepG2 should not be because NF kappa B is not expressed in those cell types. GATA should only show a footprint in K562. HNF4A should only show a footprint in HepG2. SP1 is expressed in all 3 cell types, and so it's a nice control. It should show footprints in all 3 cell types. What I'm showing you is the uncorrected prediction versus the corrected prediction. And you can see something quite drastic. The uncorrected predictions look very similar across the 3 cell types for all the different motifs. That's the red curves. And the black curves are the corrected predictions. You can see they're beautifully specific, right? Here's the NF kappa B predicted corrected footprint. It's flatlining in these 2. Here's the GATA motif has a weak footprint. And then here's HNF4A, which is like boom, bang. It just like sits there. Huge residence time, deep footprint. And SP1 has very similar footprints in all 3 cell types. Right? So this is nice. It's a good sanity check. What does it actually look at in the actual genome? So the big advantage of this model is it's predicting all these profiles at single base pair resolution at individual elements. So we don't have to just look at marginal footprints, in synthetic sequences or averaging over many sites. We can look at what they look like at individual loci. So let's go back to that example I showed you, the CDCF motif with all that garbage around it. Again, the predictions are very similar to the observed data. This is uncorrected predictions. They look really similar. Let's see what the bias model predicts. The bias model predicts this profile. And you can see something shocking. The bias model's predictions look very similar to the observed data, except in this region, right? You can see this region has less coverage in the observed and predicted signal, but the bias model predicts more spikes. Now, if you interpret the sequence through the lens of the bias model, you can see that it predicts important scores over the sequence, which are very similar to all these spurious sequences, except CDCF, right? CDCF doesn't show up in the bias model. You see some C's and G's popping up, but all this spurious signal is exact replica of what we saw in the uncorrected model. So when we do the bias correction, now we can predict the residual profile at that sequence. This is what it looks like. This is the residual signal. You see this beautiful footprint, right? Very clean footprint. And there's just the motif, the CDCF motif, and not just the CDCF motif, but you now start seeing something else, something that was hidden before. These ZBTB7A repressor binding sites starting to pop up. They're much weaker than CDCF, but they're there. They're very much there. And so you can really see kind of high resolution footprints, beautiful bias corrected motifs, even revealing motifs that were invisible before because the bias was kind of messing it up. Okay, so this is a latent underlying footprint driven primarily by the transcription factor motif syntax. And just to show you what hint attack and Tobias do, if you fit the same model to hint attack corrected data or Tobias corrected data, hint attack actually doesn't do much at all. You still see a ton of bias. It partially corrects the footprints. And this is Tobias. Tobias does a slightly better job. You can see its corrected footprint is kind of similar to ours. You see the spike that was here is largely eliminated. You create a new kind of spike here. The footprint is sort of bookending the motif very similar to ours, but it's much noisier, much spikier. And also you can see that it's not doing full correction, right? There's still a lot of background noise. The TN5 bias model we learned really beautifully cleans up the signal. And you can look at this across the genome and it looks exactly the same. It's super clean across the board. Okay. So often the question is asked, and we asked ourselves the same question. Looks great, looks like you're correcting bias, but how do you know you're not overcorrecting? Right? You could easily be overcorrecting. Clearly, there was a bias problem. We learned bias. We removed it. Looks great. How do you know you're not overcorrecting? Well, we already saw that we are learning all the motifs we're supposed to, but still, you need some other kind of independent validation, right? So luckily, we have DNA-seq and attack-seq, which both are supposed to measure chromatin accessibility. But I showed you in the first few slides, when you look at them at base pair resolution, they actually look dramatically different, right? So hypothetically, if they were doing the same thing, and if all that was different was the enzyme being used and the protocol being used, then if you had a good bias correction model, after you did bias correction, the underlying sequence syntax of the model's learn should be very similar or almost identical. And the predicted profiles after bias correction should also be much more similar than they were before, right? So that's what we see, actually. So here I'm showing you in the same cell line, DNA-seq and attack-seq. First, I'm showing you the smooth profiles. This is typically what you see in most processed data. This is like, you smooth the data and you do coverage. And this is the attack-seq p-value track from max2. You see a nice peak. You see the attack-seq DNA-seq p-value track from max2. Nice peak, right? So the peaks looks great. Agreement is fantastic between DNA-seq and attack-seq. Look at single base pair resolution and the picture is very different. This is attack-seq at single base pair resolution. This is DNA-seq at single base pair resolution. They don't look similar at all, right? There's big spikes here. Oh, look, there's a footprint right here. There's no footprint right here. What the hell happened? There's something else happening here. There's something else happening here. They look totally different. If you now use Chrome BPNet without bias correction, as I showed you before, the attack-seq is predicted very, very accurately. The DNA-seq is also predicted very, very accurately. So the model can mimic exactly what's going on DNA-seq and attack-seq separately. Now, if you do the enzyme bias correction, you learn enzyme bias for DNAs, you learn enzyme bias for attack. And now you look at the corrected predictions, something dramatic happens, right? The attack profiles, again, look really different from the observed profiles. The DNA's profiles don't look that different. There's slight correction, but they're quite similar to the observed profiles. It's telling us two things. It's telling us that the TN5 bias is immense compared to the DNA's bias. DNA's has much less bias than TN5. But after correction, you can see that broadly the shapes start overlapping really nicely. Now you see a footprint right here and a footprint right here. And DNA's tells the same story. There's a footprint right here, there's a footprint right here. Of course, the footprints are not identical for two reasons. The DNA is a tiny enzyme. It can get into nooks and crannies and give much shorter footprints. TN5 is much bigger, goes and sits there, transposase, and so it typically has broader footprints with less morphology, less fine morphology. But the real test of the model is the underlying sequence biology that it's predicting. So after bias correction, this is the contribution scores from attack from the profile head. This is the count contribution scores from attack. And first of all, they look similar, very similar to each other, which is nice because earlier they looked really different. If you do the same thing for DNA-seq, you see this from the profile head and the count head. And you can compare DNAs and attack, and they look really, really similar, right? So what this is telling us is after bias correction, the underlying sequence syntax of the two models learn are nearly identical, and the underlying footprints of the models learn are also very similar. They're not identical, but they at least highlight the exact same regions. Okay. We can quantify this again genome-wide, so we don't cherry pick some example. And we actually see this across the board. We can see that the Jensen-Shannon distance between the observed and predicted profiles for DNAs and attack, if you just compare them, without any correction, this is for the observed data, they're very different. The bigger the y-axis is the Jensen-Shannon distance, the bigger the distance, the more different they are. This is the uncorrected observed data. This is the uncorrected predicted data. And this is the bias corrected predicted data. So after you do bias correction, their profiles start looking much more similar to each other than observed or predicted uncorrected. And also if you take the deep sharp scores, the deep lift scores, the contribution scores, from the count heads, after bias correction, there isn't much of a change. Because as I showed you before, the count head doesn't learn much bias at all. After bias correction, there isn't much difference between the uncorrected deep lift profiles from the count head for attack versus DNAs, with or without correction. But for the profile heads, you see a big difference, right? Earlier they're quite different, and then you see a dramatic shift. They get much more similar after bias correction. Okay, that looks great. But as I mentioned before, let's see, I think I'm almost out of time. No, I guess I have 5 more minutes. So as I mentioned before, the footprints are not identical. They end up becoming much more similar to each other, but we can also systematically compare footprinting between attack and DNA. So here I'm just showing you again marginal footprints. These are again hypothetical footprints. You just insert the motif inside random sequence and you predict what the footprint would look like, right? So it's the marginal effect of just that motif. You can remove any confounding effects, just the footprint for that transcription factor's motif. That's another nice advantage of the model, is you can do these kinds of marginal experiments which you cannot do in the genome. Because in the genome, if you take any particular sequence, the motif is not sitting in isolation. There's other motifs sort of sitting around, and they're going to have influences on the footprints. Here you can look at the marginal influence of just one motif on that transcription factor. Sorry, on attack-seq or DNA-seq. And so again, there's really beautiful bias correction for attack-seq. You can see there's bias correction also for DNAs, but it's sort of more subtle. And you see footprints, but you can again see there are morphology differences. You can see the DNAs can get in and cut between the GADA and TAL. It's a GADA-TAL heterodimer. You can see this kind of cut in between. The TN5 is too large, it just creates a big valley. Similarly, here you got this subtle morphology. Here again, you got the GABPA, you got a little spike in the middle. It's sort of much weaker in the attack. Here's NRF1, super deep footprint, very similar, shape. There's a much sort of sharper footprint in DNA-seq and attack-seq. But the key point, and here's HNF4A as a control. HNF4A is not expressed in K562, and both of them flatline, right? So really beautiful, very clean marginal footprints. You can systematically compare DNA-seq and attack-seq now, but broadly, the two assays tell really exactly the same story. So all these claims previously made about DNAs is very different from attack-seq, and one is better than the other. It's largely experimental biases. Okay, once you remove them, we really can't make out the difference. The interpretation tracks look exactly the same, and to date we've not found any motifs that one method finds that the other cannot. And there are footprinting differences, but they are kind of very minor. Okay, they're morphologically different, but they pick up the same things. Okay, so I'm not going to get time to get into the variant effect prediction, but you can imagine doing all kinds of cool stuff with these models now for variants. And but I just wanted to highlight one key thing. I'm going to focus on the computational aspects today. We've run out of time, so I'm just going to do last 2 slides and then sort of drop off for questions. So one question you might ask is, how does this work for single cell attack-seq, right? Or pseudo bulk from single cell attack-seq? Like you showed really nice results on K562 and cell lines where you have 500 million reads. What happens if I have like 500 cells and I create a pseudo bulk from single cell data with 5 million reads? Does this fancy thing work? Well, thankfully it does. And the way we can test that out is we can take a data set with 500 million reads. We can subsample the data set, 250 million, 100 million, 50 million, 25 million, 5 million. We fit separate models on each of these, okay? And we compare to the, in quotes, ground truth, what we see at 572 million reads, okay? And so this is observed data. You can see it starts getting worse as you subsample, as you might expect. At 5 million reads, this particular enhancer has 1, 2, 3, 4, 5, something like whatever. It has very few, very, very sparse coverage. Now we look at the bias corrected predicted tracks. They look very similar to each other, right? Even at 5 million reads, it looks very similar. And what's even more interesting is if you deep lift these predicted profiles, you see the exact same sequence biology. NF kappa B with SP1 driving this enhancer. 5 million reads, 500 million reads, right? Not much of a difference. Again, we can quantify this genome wide. This is the drop in the Jensen-Shannon distance of the observed profiles as a function of depth. So we're using 500 million reads as the ground truth. As we compare them, you can see there's a dramatic drop in JSD. There's a huge degradation of the signal. Yes, why don't you go brush your teeth? Go brush your teeth. Okay, just give me 2 minutes, okay? Hold on. So this is observed data. You see a dramatic reduction in the profiles. If you look at predictions, you can see something very different, right? Like all these cumulative distributions are on top of each other, which is awesome. That tells us that the predicted profiles are incredibly similar. Only at 5 million reads, you see a bit of a drop off, but nothing compared to the observed data. So, right, so there's beautiful denoising going on. And then if you look at the deep lift profiles, you see the same thing. Up to about 10 million, 25 million reads, they are literally on top of each other. There's no decrease. There's no reduction, degradation at all in the deep lift profiles. At 5 million reads, you start losing some things, but you still can recover a lot. And if you just look at footprinting, you get beautiful footprints even at 5 million reads for most of the transcription factors. And if you look at motif discovery, again, you can rediscover most of the motifs, except at 5 million reads, you start losing some of the rarer motifs, right? This is expected. It's not magic. It's going to have some signal to noise drop, but you start seeing some drop. But it's still amazing at 5 million reads. Okay, so long story short, let me just skip to my last slide. Obviously, I have way too many slides, but video.
Given that the discussion session is next, if you would like to take 1 or 2 minutes to talk about the variant effect prediction, you're welcome to. On the other hand, we understand if you need to attend to bringing kids to school or anything like that.
There's still an hour, so maybe I could do another 5 minutes. Is that okay?
Please do go ahead. Please take another 5 minutes. That's totally fine. Yeah, thanks.
Okay, I'll just show you highlights because there's a ton of fun things to show. But let me just sorry. Okay. All right. So I'm going to really quickly run you through some examples of variant effect prediction. So now this is a profile model at single base pair resolution, which means you can do some really cool stuff. You can actually predict effects of variants on entire profiles instead of just getting some change in log fold changes of counts. You can actually see in the genome browser what the profile would have looked like at single base pair resolution if you flipped an allele, right? So we took, for example, a SPY1 binding QTL, a well-known, previously identified, very strong SPY1 binding QTL, and we pushed it through 3 models trained in GM12878. Now remember, the model has never seen variation. It's just trained on the reference genome to predict profiles single base pair resolution. And so we have a SPY1 binding model, a BPNet model trained on ChIP-seq data, encode ChIP-seq data. This is a DNase model. This is a H3K27 acetylation model. And this is the reference genomes prediction, reference allele, the C allele. You see one beautiful profile from ChIP-seq. These are the 2 strands. This is DNA-seq. This is histone acetylation. Looks interesting. Pretty decent activity. We swap the C to a G. We see this awesome, beautiful amplification of the signal in the ChIP-seq, in the DNA-seq, in the histone acetylation. You get deeper valleys and much stronger flanking profiles. And now you interpret the 3 models. They tell the exact same story. There was this weak SPY1 motif that was driving the blue profile. And when you swap the C to a G allele, it created a super strong SPY1 motif, and the 2 motifs acted in tandem and drove the signal. You can even see how these motifs act on the morphology of the signal. Notice how the blue profile has this deep dip right here in the ChIP-seq. That's sort of exactly at this motif. Okay. Remember, I'm zooming in here. I'm zooming into the center, 200 base pairs. This motif drives that valley right here, okay? When you create a super strong motif, you notice you see two valleys, you see one valley right here, and then one valley right here. The much stronger one is right here. So you can see how the variant is creating or destroying motifs, and it is changing the entire morphology of the profile, right? And the morphology of the profile is just as dramatic as the change in the read counts. So what we can end up doing is actually using a new kind of variant effect score, not just a log fold change between the counts, which is typically what people use. We can actually compute the Jensen-Shannon distance between the predicted profiles for the reference and ordered allele. So we're looking at the change in the shape of the signal. And this can be very powerful because you can see very subtle effects of motifs and variants on long range, profiles across the genome, right? So I love this result because it shows that sometimes you do not need ultra complicated models. Chrome BPNet is not complicated, to be honest. Very simple convolutional model. You can train it in about 3 to 4 hours on a single GPU, on your laptop, no problem. You train on the entire genome. You get a really high performance model. It will do bias correction for you. I'm just comparing it to a few strong baselines. One is delta SVM, which was the previous state of the art, on some DSQTL data. And I'm also comparing to Informa, which is this recent model from DeepMind and Calico. It's a giant transformer. You cannot even train it on a regular GPU. You need Google TPUs. Very, very expensive, takes a few weeks to train. And it's a multitask model. It's trained on many, many readouts at the same time. Our Chrome BPNet model is very modest. It's just trained on one data set, the GM12878 reference data, no multitasking, does bias correction, and we convincingly beat Informa by a big margin. Okay, the DNAs and the attack-seq heads beat Informa by a big margin. This is the benchmark from the from the Delta SVM paper. So what we try to do is we try to run our models on other people's benchmarks so that we don't cherry pick our benchmarks. If we can beat them on their benchmarks, I think we're good. And then we, of course, have our own benchmarks and we compare their models to ours. And that's a good practice in general, because you can always find a data set where your model does better. It's a little harder to beat people on their benchmarks because they optimize their models on their benchmarks. So this is the Delta SVM benchmark. We definitely beat Delta SVM by a huge, huge margin. But even for Informa, that's the gray and the red lines. There's still a big boost that we get from Chrome BPNet. Okay. So bigger models are not necessarily the best. If you're careful about how you model stuff, you can beat big models with small models. Again, on MPRA, saturated mutagenesis from the Informa paper, this is Informa's benchmarks. These are predictions from our models. These are predictions from Informa. These are correlations between the observed and predicted MPRA effects. For most of the enhancers, we are better than Informa. For a few, Informa is slightly better. But you can see there are dramatic shifts in correlation between Chrome BPNet's predictions and the observed data versus Informa's predictions and the observed data. So I can guarantee you, the paper's not published, the code is, but if you compare us against Informa on any data set, you've done this on tens of thousands, we are substantially superior to them with no loss of accuracy and substantially reduced computation load.
And you attribute that to the bias factorization, the bias correction? Is that essentially, yeah, what do you attribute that to?
Two things. So the first thing is we train on single base pair resolution profiles. There's a huge, huge advantage of doing that. The reason is because when you try to predict a single scalar from a sequence, there are many ways in which the neural network can make that prediction, right? There's a lot of instability. We've seen this for years, okay? We've not done variant effect predictions ourselves for years using neural networks. We, in fact, had a paper that was published in Nature Genetics where we used SVMs after a student of mine spent like 2 years fitting neural networks on that data because we found that if you trained 5 folds of the model, they disagreed with each other like crazy. They were like super unstable. As soon as we switch to single base pair resolution prediction, the profile shapes, right, are very constrained. They're exactly mimicking what you see in the sequence. So the models learn incredibly stable models, and the shape of the profile tracks the sequence syntax so well that you learn the sequence syntax also extremely well, right? So coupled with base pair resolution prediction and bias correction, you get incredibly, like, really powerful models. I would love to see somebody like really beat ChromeVPNet by a big margin. I think it's going to be really, really hard. We've tried all kinds of stunts and even shoved a transformer into the small model. Not much movement, right? So I think we are really kind of hitting that ceiling. There's still a lot to do, but that's a different question.
Yeah. No, it's an amazing conclusion and message, especially in the age of huge models now. So
yeah, so our goal is to continue building models that we strongly believe that for genomics, we do not need, scale is everything. I think if we're clever and careful, we can continue training small models that will continue doing very well. Even for expression prediction, which is supposed to be a really hard problem, we don't have a solution yet, but we have ideas and we're trying to build small models so that you can continue training on regular hardware. Right?
And do you believe that beyond the tractability that there's an advantage for interpretation and for sort of interesting models understanding what they're I figured you
absolutely. I showed you like interpretation is trivial, super stable. You go to transformers and stuff, all bets are off. There aren't really good ways of knowing how to interpret transformers. All our current methodology works beautifully for CNNs, right? Just like super stable, works very nicely. So the big advantage of staying small is interpretation. If that's the key goal, it has a big effect. So anyway, there are lots of other things I don't have time to show you, but we can predict, CAQTLs really nicely. We can predict across population.
Anshul, sorry, just a quick other question. And I mean, amazing science as usual. That's awesome. So your models are trained on data from how many donors? And could you comment on what your thought is about training models on, natural genetically diverse data sets, deep sequencing data sets?
So our model is trained on one sample.
Right.
No training across many.
So the model doesn't see the genetic variation at all.
It doesn't. It just uses you can give it genetic variation, no problem. You can give it a personal genome, but we just currently give it the reference. We've also given it personal references and it works fine. It looks very similar. But just to show you the proof of the pudding, what you asked, here's an example where we trained a model on the GM12878 European LCL line, right? And we're predicting QTLs derived from individuals from very diverse African ancestries, San and Yoruba and all these. So it's a complete cross-population prediction, and we see almost no drop. It's very, very marginal. It's mostly batch effects. There's a slight batch effects that happen, but you can see the numbers right here. It's 0.93 and 0.88, right? So if you predict within African populations, like you train a model on the African data and you predict on the African data, you get certain correlations. And if you train on the European model and you predict in African individuals, you barely get any drop, right? So this is telling us that the underlying biology between Africans and Europeans is the same. And so if you have a model that's learning the underlying syntax well, it should generalize very nicely. And that's what we're seeing, okay? I'll skip that, but we can apply this to colorectal cancer, single cell data, organoids, lots of fun stuff.
Oh my God, we're going to have you back for another. Go ahead. Sorry. Go ahead, Melina.
No, just a quick question. I think I've seen the data. So you looked at posterior probabilities from multi-ancestry GBAS data and how the model predicts, causal regulatory active variants and their relationship to posteriors.
Yes, I'll show. Yeah, that's in the last slide. 1 second, let me show you.
Sorry, I'm extending. I'm extending you beyond.
So this is comparing to fine mapping data.
Yeah.
I love this slide for 2 reasons. This is a model trained on mouse. Okay, firstly. So we're taking a model trained in single cell ataxic of coronary smooth muscle cells, coronary vessel smooth muscle cells from a atherosclerosis mouse model. Okay, we got beautiful data in the mouse, and we can track, atherosclerosis temporally. So you get all these amazing cell states which you cannot get in the humans because it's very hard to get that kind of temporal information. This is in actual mice with lineage tracing. Okay, it's unpublished again. So we're training a model in smooth muscle cells in a specific cell state in mice, and we take that model and we predict in the human genome, right? So now we're taking a whole bunch of heart GWAS variants. This is a bunch of traits, heart-related traits, systolic pressure, blah, blah, and there's really nice GWAS data, and there's fine mapping from Hillary's group, right? So what we're showing is as a function of posterior probability of fine mapping. So we're going from low posterior probability to high posterior probability. So we're changing the PIP threshold, and we're changing the Chrome BPNet threshold as well of predicting, is my SNP functional or not, right? And what we're doing is we are predicting, so we're making predictions and we're comparing the observed to expected overlap between Chrome BPNet predictions and GWAS fine mapping. You can see as you improve the PIP threshold, you see these absolute skyrocketing enrichments, right? It's like 20, 40, 60, 80, 100-fold enrichments.
Yeah.
So we are doing, like, this is a mouse model in humans. We have similar results for human models in humans when the cell types are well matched, but this stuff is actually working now. It's doing pretty well.
So is it, yeah, that looks crazy. That's amazing. Do you improve that even further with context specific data sets? Human,
Yes, it's super important. Like we have
Yeah.
the same result in the coronary smooth muscle atherosclerosis time course. We have many cell states. It's smooth muscle cells, going into a fibroblast-like state. And so we have a continuous trajectory. We can sample across the trajectory. And there is a specific cell state where this skyrockets.
Yeah.
It's not across the board. Okay.
That makes
It's
a lot of sense.
Intermediate, it's a chondromyocyte state or something like that. And that's where it really skyrockets. I mean, you still see enrichments in the original smooth muscle cells, but it really, you start seeing it in specific cell states.
Super. Thanks, Anshul.
Yeah. And this is what it looks like. You can like look at individual loci and you get these beautiful allelic effects. And, you can see consistent effects in vivo mice, in vivo humans, and even in cell lines where you might want to test the variant. So we can actually make predictions about which cell lines are a good fit for the data. Anyway, it's a lot I've said already. So if you want to test Chrome BPNet, the beta is out. Paper hopefully will come out in a month or so, but give it a shot. We've spent a lot of effort making it very usable. There's lots of tutorials and so forth. Just go to Kundaje Lab, Chrome BPNet. Feel free to use it. I think it's pretty stable, and we've got good feedback from other folks using it too. So give it a shot and let us know what you think. And with that, I'll just stop. Again, thanks to Anushree and Anna. It took us 2 weeks to fit the first uncorrected model, and took us 1.5 years to get to the corrected model. That's the time gap. And again, thanks to a lot of other people in the lab doing really great stuff on all the variant effect prediction and, of course, funding sources.
Anshul, thank you so much for the amazing talk on spectacular work. These are really exciting. This is amazing progress in this area. There was already, the existing models were already doing quite amazing things, but this seems like a big step forward.